Hello students! Today we are going to get into the details of the process of mitosis and cell division. Recall from last time that individual cells have life cycles composed of different stages. The longest phase, interphase, is the time when the cell is growing, developing, and performing the activities of daily life. Sorry about the bell. However, at some point, the cell will want to divide to make more cells. So it will copy its DNA and its organelles, and then it will move out of interphase and into M phase. The first part of M phase is mitosis, which is the process of dividing the cell's original nucleus into two new identical nuclei. We stated last time that mitosis happens in 4 stages, and they are called prophase, metaphase, anaphase, and telophase. Today, we will begin with prophase. So, let's remember that a cell that is entering prophase is coming out of interphase. So that's why I've got this picture here, because it's always good to remember which phase you're coming out of so that you can see the changes that occur in the new phase. So, a cell comes out of interphase and goes into prophase. By comparing both these pictures below, you can identify some of the important events that occur in prophase. First, notice how during interphase, the chromosomes are loosely unwound into a form called chromatin. On your picture here, I'll write down chromatin. Okay, and here it kind of looks like spaghetti, but those are chromosomes, but they're loosely unwound. However, in order to prepare to separate the chromosomes precisely with the right number and the right type going into each new nucleus, it makes sense for the cell to consolidate and organize the long strands of DNA by coiling them up and condensing them into chromosomes, like we saw in our last chapter. So you can see that here in prophase, we've got our condensed duplicated chromosomes. Then, to further keep them organized, each chromosome gets connected to its duplicate. Okay. And that you can see in the pictures, we've got the 2 chromosomes, and in this stage we're going to connect them together so that they stick together when we're trying to move them around the cell in order to separate them. So we've got this structure. Recall that the center of the structure is called the centromere. And that the centromere the centromere holds together the 2 identical chromosomes, and when they're in this form, either one of the chromosomes, we now call them sister chromatids. And I am spelling things incorrectly. So these are called sister chromatids, and I will refer to them when they're in this configuration, stuck together by a centromere, by this name. Okay, so the next thing that happens during prophase is that the nuclear membrane dissolves, like we see right here. This must happen because we are going to move the chromosomes out and around the cell so we can end up putting them into two different nuclei. Finally, during prophase, two organelles called centrioles assemble themselves and move away from each other to opposite sides of the cell. So that is what we see happening right here. These centrioles are moving away from each other, and we're going to see why this is important that these centrioles are moving away from each other when we get to the next stage. Okay, now we move from prophase into metaphase. In metaphase, our duplicated chromosomes, which are connected at the centromere during prophase, will now line up in a very orderly single file line in the center of the cell. Remember, it is very important that the number and type of chromosomes stay organized so that each new cell gets a complete and correct set of information. Once our chromosomes are lined up, rope-like structures called spindle fibers are organized by the centrioles to stretch from either side of the cell and attached to duplicated chromosomes. So you see those there. Those lines are the spindle fibers, and you can see them attaching at the centromere of each set of chromosomes, or of the set of sister chromatids. At this point, the cell is done metaphase, so it will move to the next phase, which is anaphase. Okay, so here's our metaphase cell transitioning into anaphase. By looking at these two cells, you can see that during anaphase, each original chromosome lined up in the center of the cell is separated from its duplicate. After they are separated, spindle fibers organized by the centrioles at each end of the cell will begin to pull the separated chromosomes to opposite sides of the cell. So we can see that here are identical chromosomes. They're separated and they're moving in opposite directions. Another thing you can also see happening here is that the cell is elongating in shape. So it's taking on a more oval shape in preparation for the last phase of mitosis, which is telophase. Finally, anaphase transitions into telophase. During telophase, we finish mitosis. Now remember, mitosis is the process by which a cell nucleus is divided into two new nuclei. And so here, we should notice that we are indeed ending with two new nuclei. During telophase, the spindle fibers will disappear because we don't need them anymore. In addition, nuclear membranes will reappear around each new nucleus. So we can see that occurring right here. Each nucleus becomes redefined by having a nuclear membrane wrapped around each set of chromosomes. Another thing that happens during telophase is that chromosomes begin to uncoil. Now, that is not shown in this picture, but you can imagine what the chromosomes looked like in interphase, how they were in the loosely unwound chromatin. And so that process of unwinding will begin here in telophase. Finally, the cell itself, which is now very elongated and oddly shaped, is going to pinch in the middle. This is to prepare it for the next part of M phase, which is cytokinesis. Okay, we have successfully described the division of the cell nucleus and therefore have finished describing mitosis. You'll notice, however, the cell cycle is not complete, as we have 2 nuclei, but not 2 individual cells. We will complete our explanation of the end of the cell cycle next time, so stay tuned. Don't forget your Quizmodo, and have a great night.